Hello and welcome to the webinar. Today we will talk about how to best profile the long noncoding RNA or lincRNA expression. The major motivations of profiling the long noncoding RNAs are the functional roles in the regulation of protein coding genes by various mechanisms, the associated biological functions in human diseases, and the higher specific expression patterns for biomarker applications. The fundamental difference between mRNAs and lincRNA is that lincRNAs do not encode proteins. Beyond this, there are many similarities. For example, both lincRNAs and mRNAs are greater than 200 nucleotides in length, often undergo alternative splicing to produce transcript isoforms. Some lincRNAs, but not all of them, are polyadenylated. These similarities mean lincRNAs and mRNAs can be technically profiled simultaneously together. However, there are also many properties that are different between lincRNAs and mRNA, which pose special challenges and require special considerations. For example, lincRNAs do not encode proteins. The annotation and the analysis of profiling data are different. LncRNAs are more likely to act at an RNA level by complex genomic contexts in either sense or antisense strand directions. Also, lincRNAs are mostly in the nucleus, whereas mRNAs are mostly in the cytoplasm for protein translation. LncRNA as populations are expressed and functionally operate at much lower abundance levels than mRNAs. Based on the analysis of gene code catalog, the mean expression levels of lincRNAs is about 1/10 of the mRNAs. Many lincRNAs are present at only a few copies per cell. For example, in the single molecule RNA fish or fluorescent in situ hybridization in the picture in the middle, N-real lincRNA is expressed at no more than 2 copies, as compared to many copies with FOXF2, F1 mRNA. The other reasons of lincRNA being low in abundance is lincRNA expression can be highly specific. Even in the same tissue, only the specific cell type may express that particular lincRNA. As in this example, in the picture on the right, different lincRNAs are specifically expressed in different cell types in the hippocampal region. Each lincRNA in the found histological structure may be diluted in the overall tissue mass and diluted in the overall total RNA sample. LncRNAs should not be dismissed for their expression at low levels. Many of them do functionally operate at low levels. Some often observed mechanisms of lincRNA regulation on neighboring genes are the latently transcribed lincRNAs tethering and recruiting protein complexes, such as chromatin modifier PRC2 to modify local chromatin active state, or spliceosome to facilitate ongoing transcription, or acting as enhancer RNA to form chromatin enhancer loop. Unlike diffusible factors that act in trans, the cis-acting mechanisms do not need the lincRNAs to be present at a high solution concentration. For example, hot-tip lincRNA is expressed at 1 to 2 copies per cell to functionally control the Hox gene cluster. Another group of enhancer lincRNAs or eRNAs activate target genes, and yet their abundance are generally very low and difficult to be detected by RNA sequencing. The lower the lincRNA abundance poses significant numerical challenges to lincRNA profiling by RNA sequencing. In RNA sequencing, with ribosomal RNA depleted RNA, the top 1 high expressor genes, such as the housekeeping genes, occupy the overwhelming 70% of all sequencing reads. The modestly expressed mRNAs contribute 30% of the sequencing reads, whereas lowly expressed genes such as lincRNAs only receive about 1% of the sequencing reads. Based on some randomly selected actual RNA sequencing mapping results, 40 to 80% of the RNA sequencing reads can be mapped to the mRNAs, whereas only 4 to 12% of the sequencing reads are mappable to lincRNAs. In the picture on the left, briefly, the theoretical quantification accuracy, or on the other side of the coin, the error, is related to the read count coverage by Poisson model. That is, the lower the read counts, the higher the error, as depicted in the blue curve. The actual observed error is much larger, as shown by the orange curve. This is well known as the over-dispersion. Typically, to achieve a reasonable accuracy, at an error less than 20%, more than several hundred of the reads are required for the transcript. In the picture on the right, in a typical 40 million sequencing depth designed for mRNA sequencing, the sequencing read coverage for lincRNA fraction is very low. Less than 10% of lincRNAs can be quantified reliably. In other words, more than 90% of lincRNAs cannot be reliably quantified. For microarrays, the oligo probes on the solid phase are able to affinity capture and enrich the rare target sequences, while at the same time measure the fluorescence intensity as the abundance levels. Therefore, microarray accuracy is less impacted by the transcript at the low abundance levels, as shown in the red curve, compared with RNA sequencing, shown in the black curve, in a very large clinical study published in PNAS. The other important aspects of lincRNA profiling is that lincRNAs are often produced in alternatively spliced RNA isoforms having varying, sometimes opposite functions. For example, MEX3 gene produces more than 10 MEX3 lincRNA transcript variants having various p53 tumor suppressor activating activities. So it's important to profile different lincRNA transcript isoforms. However, as shown in the picture on the left, there are inherent challenges of using next-gen sequencing to profile gene expression at a transcript level by sequencing. That is, the short RNA sequencing rates, the low lincRNA abundance, and even lower transcript isoform levels, receiving diminishingly low sequencing rates. The computational complexities in dealing with the alternative splicing are also daunting. As in the picture in the middle, using the top sequencing aligners, HiSet2, TopHat2, and MapSplice2, these commonly used in RNA sequencing data analysis, the sensitivity of detecting both mRNAs and lincRNA isoforms assisted with the reference transcriptome is less than 20% recall. In the picture on the right, to find new transcript isoforms, de novo transcript assembly from the sequencing rates is required. Even the top de novo assemblers perform poorly for the transcript isoforms and particularly poorly for lincRNAs, with only a few percentage being detected. In fact, even with the same dataset, different algorithms and softwares produce highly variable exon models for the transcript isoforms and very different profiling values. To resolve these issues more effectively, a newer generation of long reads NGS is needed. These transcript-specific profiling difficulties are highlighted by the paper published by Sequencing QC and Microarray QC3 Consortium in Nature Biotechnology, even for mRNA. That is, for genome-scale RNA sequencing, this is particularly difficult because of the sampling noise from low read counts for many transcripts. Again, in another paper published in PNAS, the same conclusion is reached by a very large and deep RNA sequencing study. That is, 300 million fragments are required for the detections of a specific human alternative splicing event with 80% power. Compared with RNA sequencing, Arraystar LncRNA Microarrays use the array probes specifically targeting the non-lncRNA isoforms. The transcript isoforms is constructed from the data sets at a species level. Many of the transcript models were constructed with the experimental support, including full-length cDNA sequences. The splice junction-specific probes, or unique exon probes, ensure straightforward, sensitive, unambiguous, and reliable detections and quantification of the transcript isoforms. As an example, the BCL2L1 gene has BCL-XL, BCL-XS, and ENST transcript isoforms, having opposite oncogenes or tumor suppressor functions. Each of the transcript is detected by a unique probe. In addition to the expression values, the annotation and data analysis are integral parts of the lncRNA expression profiling. Unlike mRNAs that have been well curated and compiled in the major databases such as GenBank, lncRNAs are more scattered in many specialized databases. Large number of lncRNAs discovered in recent studies have not been entered in the major databases. Arraystar LncRNA Microarrays continuously and comprehensively resource lncRNAs from many data sources and lncRNA study publications, including Arraystar's own lncRNA collection pipeline from more than 47 terabytes of data in the current microarray version. It is true that lncRNAs have been less studied than proteins. Nonetheless, a growing number of lncRNAs have been better annotated than just anonymous RNA sequences. These full-length lncRNAs may have been functionally studied, experimentally supported, and described by at least one PubMed index research paper. These lncRNAs receive gold standard lncRNA status and are indicated in the annotation for the available study information and as low-hanging fruits. Arraystar Human lncRNA Microarray version 4 has the best contents with more than 40,000 lncRNAs and the entire mRNA encoding proteins cataloged by UniProt. Among the lncRNAs, more than 7,000 are gold standard lncRNAs and 32,000 are reliable lncRNAs. Arraystar LncRNA Microarray comes with rich, detailed lncRNA annotations and analyses, which are integral in the lncRNA profiling, essential for the understanding of the biological function and significance. lncRNAs are systematically annotated, representing the most recent advances in the field. lncRNAs are broadly classified into enhancer lncRNA, or eLncRNAs, transcribed from the genomic regions with enhancer epigenetic marks, and the promoter lncRNAs, or pLncRNAs, from a promoter epigenetic mark. The genomic relationship with the closest labeling coding genes, such as intergenic, divergent, antisense, sense, intronic, serve important clues as to what are the target genes and how these targets may be regulated. Starting from the second row, the 5' ends, or the full-length lncRNAs, are indicated by the cap analysis gene expression sequencing, or cage sequencing data that can be pointed to the precise location of the lncRNA action. A subcellular location of the nucleus or the cytosol helps to predict lncRNA functions. A nuclear lncRNA is more likely to be involved in transcription or nuclear RNA processing, whereas a cytosolic lncRNA is more likely to act on cytoplasmic functions such as mRNA translations or competing endogenous RNAs for microRNAs. Certain important subgroups of lincRNAs, such as intergenic lincRNAs, lncRNAs, antisense lincRNAs, competing endogenous RNAs or cRNAs, or the lncRNAs as enhanced RNAs, or natural antisense RNAs for target mRNA masking, interference, genomic loci imprinting, and methylation, or cRNAs for microRNA-mediated gene regulation are subgrouped. The lincRNAs having high conservation among the species, such as the transcript of the ultra-conserved regions, or TUCRs, may underlie vital functions indispensable for the organisms. LncRNAs are known to associate with diseases, biological pathways, or processes, or highly specific expression patterns in tissues are compiled and annotated. The expression patterns of the lincRNAs and mRNAs on the same chip allow positive or negative correlations of the genes for the up or down regulatory relationships. The correlated protein coding mRNAs are a bridge to the lincRNA gene set analysis to indirectly predict the biological functions of the lincRNAs. Further, the lincRNAs and mRNA regulatory interactions can be graphs in the coding non-coding co-expression network, or CNC. This table highlights the major reasons why Arraystar LncRNA Microarray is the technology of choice for profiling the lincRNAs. You may pause the video to read the table. One known scientific thinking is that next-gen sequencing is somewhat new and novel. The fact is that next-gen sequencing has been around for more than a decade. It's not much novel nowadays, so use the best tool for the purpose. Arraystar LncRNA Microarrays have been powering the lincRNA research for many years, producing high-impact, groundbreaking results. For example, as published in Nature Cell Biology, Arraystar LncRNA Microarrays were used to profile the total cellular RNA T and RNAs bound to the lipid membrane L from the normal and the triple-negative breast cancer tissues. A differentially expressed lincRNA, lincA, was identified from the normal and the triple-negative breast cancer tissues. Native lincA can be pulled down from the lipid binding. Likewise, the recombinant lincA binds to the lipid membrane dot plot. The binding affinity to phospholipid PIP3 is very strong in the nanomolar range. In breast cancer, lincA gene contains certain SNPs that are amplified and are highly expressed, indicating its relationship with breast cancer. lincA overexpression promotes cancer and confers anti-cancer drug resistance by hyperactivating serine threonine kinase AKT while binding to PIP3. Published in another paper, Nature Cell Biology, lincA interacts with and phosphorylates hypoxia-induced factor HIF1-alpha, which in turn activates histone acetyltransferase P300 to turn on the target gene promoters for cancer metabolism and glycolysis. lincA is identified as a missing link in triple-negative breast cancer. The work is very significant in the field. It is selected as the editor's choice in science signaling. In the picture on the left, published in Cell from the SciLab, using Arraystar LncRNA Microarray, BRARS4 lincRNA was identified differentially expressed between breast cancer and the pre-tumoral tissues. In the picture on the right, upon cytokine stimulation, BCL4 binds SNP1 and PNOTs to relieve their inhibitory effects on the histone acetyltransferase P300 and RNA polymerase II respectively, which promote the transcription of cytokine CCL21, sonic hedgehog transcription factor GLI2 downstream target genes that are responsible for cell migration. In the picture on the lower left, ectopically expressed BCAR4 promotes cancer growth in xenografts model. As an example of lncRNA cis-activating oncogenes, GC-link1 was identified on the Arraystar LncRNA Microarray as a differentially expressed lncRNA. It was annotated as exon sense overlapping with SOD2 gene. Further studies, in fact, have identified GC-link1 binds to two activating chromatin modifiers WRD5 and KAT22A, which leads to activation of SOD2 gene, a gene for cell death protection and gastric cancer metastases. Hundreds of papers have been published using Arraystar LncRNA Microarrays. These slides list some of the publications published in leading journals. So for summary, for global lncRNA profiling, Arraystar LncRNA Microarrays are the superior choice. I will stop here and thanks for watching.